Okay. Hello, everyone. Good afternoon, everyone. Good morning, good evening, depending on where you are coming from in the world. I know that we have audiences from all over the world, and obviously the times are not the same. Thank you so much for joining us, and welcome to the 8th edition of the GSU Malaria Journal Club, which is a monthly series. Today's webinar is a vector-focused one, which is going to reference Odero et al.'s recent preprint, which is titled Discovery of Knockdown Resistance in the Major Malaria Vector Anopheles Funestas Reveals the Legacy of Persistent DDT Pollution. My name is Pablo Chauke, for those of you that do not know me, and I'm a training coordinator at Malaria Gen, working with an awesome team of people to organize these monthly events. Just a few ground rules. The Journal Club is being recorded and will be available on our YouTube channel later today. Please kindly stay on mute. We will have a Q&A session at the end where you can raise your hand or type your question or comments in the chat or Q&A box. So please, while Joel is giving his presentation, do comment or ask your questions in the Q&A, which will be addressed at the end of his presentation. Without further ado, I would like to introduce our speaker for today, Joel Odero, who is a busy traveler. Just last month, he was at MIM conference in Rwanda, where he gave an amazing presentation there. He's a PhD candidate. He's a researcher. He's a teaching assistant on the current PAMCA Malaria Gen training course, which is currently running, and so much more. So please do talk to him at the end if you want to learn more about him. The title of today's talk, as you can see on the screen, aligns with the preprint. Just a short bio about who Joel Odero is. Joel Odero is a vector biologist and research scientist at the Ifakara Health Institute in Tanzania. He has a conviction for fighting vector-borne diseases and is building his research career at the intersection of field, lab, and the application of genomic tools to capture and interpret biological data. His PhD training at the University of Glasgow integrates field mosquito ecology and population biology, giving him a unique combination of skills in vector research. They have recently discovered knockdown resistance, or KDR for short, mutations in Anopheles funestas, the dominant malaria vector in East as well as Southern Africa, marking the first detection of this resistant mechanism in this vector. Joel aspires to lead an interdisciplinary group at the cutting edge of research on vector biology and public health intervention with a long-term plan to develop sustainable technologies to alleviate the global burden of vector-borne diseases. Joel Odero, thank you so much for joining us. You may begin your talk.
Thank you, Pablo, for that great introduction. I'm gonna now go straight into the talk. I'll be speaking about a recent preprint that we had out about the discovery of KDR in Anopheles funestas in Tanzania. Just before moving from this, I should mention that Anopheles funestas is one of the major vectors, or the major vectors for this case in Tanzania. So just keep that in mind on why we are interested in this vector. Okay. So I'll start with this, probably a very common kind of slide in most talks, but as we might all know as a community, is that progress in malaria control has really stalled, and there's both in terms of malaria deaths and cases increasing over the years, and there's something that is concerning and a focus of many programs on how this can be averted. But mostly for malaria control, it's very reliant on vector control. And I just put this snippet here from Samir Bhatt's paper, showing us that vector control using mostly bed nets and IRS, that's spraying of houses with insecticides, has really contributed a lot in reducing malaria, especially in sub-Saharan Africa. But as we both know, as we all know here, that these tools are insecticide-based. And being insecticide-based, one of the main challenges with this is obviously insecticide resistance. So vector resistance has really increased in Africa. And this just shows the evolution of resistance in Africa across the years. And looking on the map, we see that from 2005, you don't see much resistance. And resistance in this is being shown by the strength of the colors here. So the more dark the color, the higher the resistance. But as we move across the years up to around 2017, when this data set reached, you see that the dark parts are really increasing across Africa. And this shows on how resistance has really quickly evolved across this, and mostly in response to the vector control tools I just spoke about. But I think it's usually important to highlight this because this is data, modeling data from Anopheles gambi. And the reason why you don't get like all vectors in this, especially Anopheles funestas, is due to challenges with doing bioassays for Anopheles funestas. And this includes getting like the premature stages to do the bioassays. So how does insecticide resistance evolve in vectors? I'll keep this very short, but usually when you expose a naive population to an insecticide, they'll be pre-selected for this. So those that can survive the insecticide will then pick up that, and then these alleles that enable them to survive the insecticides will then quickly spread in the populations, and you end up with a population that is selected as strongly resistant to the particular insecticide that you are subjecting them to. But mostly the resistance mechanisms will involve steps that can be like increased excretion of the insecticide in the mosquito system. It can also be the increased detoxification. So insects will have a detoxifying mechanism, mostly the cytochrome P450 systems, that allows the insect to get rid or to clear out the insecticide from their system. But there is also another mechanism which is very important for most malaria vectors, which is a target side modification. The target side for insecticide insecticide on the mosquito is usually the voltage-gated sodium channel, the VGSC in short. So the VGSC usually when you have is like the VGSC, this a structure of the VGSC usually like has 4 domains, each with at least 6 subunits. And in this, when you have like mutations on the transmembrane of this can easily cause what we call what we call a target site mutation. So usually in mosquitoes that don't have any mutations, insecticide will bind to this site, and this disrupt the ion conducting state of the VGSC, and this causes paralysis, or what we call knockdown. But because this is specific, highlighting the different mutations for Anopheles gambi, and in this, the red dots here indicate the known mutations that have been known to confer resistance in Anopheles gambi. But the most important mutation here is the 995, which is on domain 2 subunit 6. And this has been shown to cause very strong resistance in Anopheles gambi, Anopheles colusa, and in even some other insects. So KDR frequencies or knockdown resistant genes, this usually they appear in the population in response to insecticides, and their frequencies pick up quite fast. So this is an example of this from some work that was done by Natasha some years back. And you see in both the intervention and control arms, when they when they put in an intervention, the KDR mutations start to peak. But in a very short time, in a matter of months, this mutation, both in the intervention and the control arms of their trials, you find this KDR mutation picking up quite fast. And a good demonstration, sorry, a good demonstration of how KDR will affect resistance to insecticides is what we see here. The plot on your right is as KDR frequencies increase in a population, then the resistance to, in this case, a pyrethroid, also it increases. And there's a direct correlation between KDR and resistance to insecticides. So how about the evolution of this? So this is one of the initial works that was done on KDR, just looking at the different KDR haplotypes across Africa. And what we see here is KDR haplotypes appeared and they quickly spread across Africa. And this work done by Chris Clarkson shows that most of these KDR haplotypes, especially in the west of Africa, they had like a single origin, and this single origin then quickly spread across most of West and Central Africa, and the same is also seen in the east of Africa. So essentially, it means that KDR really wipes so quickly in populations because it's so important to the mosquitoes. So then coming to the Anopheles funestus. So Anopheles funestus has been for a very long time thought to not have KDR, and there's been a substantial amount of work done on this. And I think probably the most important work on KDR in Anopheles funestus come from Charles Wodge's group, where they use a pyrosequencing method to look at KDR in mosquitoes across Anopheles funestus mosquitoes across Africa. And they did find some mutations in these mosquitoes, but importantly, they didn't find mutation in the 1014 codon or the 995 codon in Anopheles gambi. So after this, I think most people have been referencing to lack of KDR or 995 or 1014 KDR in Anopheles funestus. But what we recently were doing some surveillance work in Tanzania. So we were doing both phenotypic surveillance and also genotypic surveillance. So we were exposing Anopheles funestus mosquitoes to different doses of insecticide that are used for vector control in Tanzania, including some pyrethroids. But when we exposed mosquitoes to DDT, we noticed a very unique pattern of resistance or DDT resistance that was very particular to a site in the Kilombero Valley in Tanzania. And I should mention that DDT resistance is quite uncommon in most of East and Southern Africa. And this is the reason why DDT has been used in the past to try to either reduce or eliminate malaria in most of these countries in the southern of Africa. But again, also DDT is an insecticide that, yes, was banned in the past, but has always been used and pre-qualified by the WHO for vector control, I think up to about 2017 when it was delisted. So then once we saw this very strong resistance here, we kind of got thinking on this, and what we did then is to do some whole genome sequence for samples again collected across Tanzania. So these samples were sequenced at full coverage, and then were aligned to the new reference Anopheles reference genome from Mara's group. And also, I should mention that these samples were processed through Malariagen. So one of the unique things that Mara's group observed was this presence of a unique selection peak in the area where you find the VGSE. And this was very particular to samples from Tanzania. And in particular, Morogoro, which was in their data sets. So for then the samples that we sequenced, these are about 333 samples that were sequenced at full coverage. When we did a selection scan at this specific region where you expect the VGSE to be, that is on chromosome 3, what we saw was a very clear signal of G123 here, which is a measure of selection in Morogoro. And this was something that we did not see anywhere else. There was some signal that we also saw in Ruvuma, but very not very strong signal, and also some signal that we saw in other locations, but these were in very like not of clear signals though in so then we checked on the frequencies of the non-synonymous alleles in this case. So how we did this is to look at our top non-synonymous genes, and these are the genes that can cause a change in protein function. And for this, we picked up eight of these genes, and in the 8 of them, we found that the 976 and 1842 had very high frequencies in particular populations. So the populations in here were in Pwani. So Pwani is in the coast of Tanzania, in Morogoro, in the southeastern, and also in Ruvuma, which is in the south of Tanzania. And these were frequencies that were ranging from for 976, these frequencies could go up to 90% in some populations. But you realize here in Morogoro, we had samples, historical samples, so samples that were coming from the way back to 2017 up to about 2023, that is last year, the samples that we sequenced last. And you see, like across the years, the frequencies of both mutations are quite high, and quite high, I mean, at least they're about 50% for 976, and the same for even 1842. But when we checked for these frequencies over the years for this for the site in Morogoro, we saw that there was a decline in the frequencies of both our top mutations here over the years. So then we picked up the top mutations and we wanted to investigate this further. And one of the things we did is to align these these sections to Anopheles gambia and Musca domestica sequences. When we did the alignments, we found that for 976, this was corresponding to 995. So 995 is similar to 1014. So 995 is the numbering for Anopheles gambia. So, and again, 1842 was similar to 74 and 79 and 1879 for Musca domestica. And this was a clear indication that the mutation that we were seeing, the 976 mutation that we were seeing in Anopheles funestas, was indeed a non-KDI mutation, which is similar to what you see in Anopheles gambia. But usually these mutations, just like in Anopheles gambia, they occur in pairs. So we did then a linkage disequilibrium analysis for this to see on how the two top mutations co-occur. What we found is the two mutations were really tightly linked, and this was up to around 95% of the time they'll co-occur. And for 995 in Anopheles gambia, this is a mutation that is known to occur with other similar mutations, such as the 1570. And when this does happen, it increases then the resistance phenotype that it confers in the mosquito. Okay, then one thing we are also interested to see is the kind of the history, the check a bit of the history of these mutations. And one way you can do this is to do haplotype clustering. So for this, we had 666 haplotypes on this. And so down here, we have the populations. So these populations correspond to the 9 different collection points that we had. And we also have the non-synonymous SNPs. So the 1842 and 976 up there. And the tree there, each of the nodes on the tree represent single mosquitoes, the 333 of them. So from this, we can see that all samples from Morogoro, which is kind of the pink there, they all in a region where you have a strong selection or selective sweep in this case. And so all the samples are there, and the only other samples that come up are around here, or also show like presence of the 976 and 1842 are samples from Pwani and also from Rovuma. But I think the point here is that the populations from Morogoro that has that has the highest frequency of these mutations, that they had the same haplotype, all of them. So this is probably a signal that that this is kind of the mother haplotype, and this is what is then spreading to all these 2 other locations where we see the KDR happening. So I think importantly also is wanted to do like, okay, we've found these mutations. So what do they then mean to the pesticides that are being used for control? So the way to do this is we got mosquito populations from the field, exposed them to deltamethrin and also DDT. These are diagnostic doses, so doses that you'll usually use in the WHO bioassays. And at the end of 24 hours, we separated those that were alive and those that were dead, and then amplified and sequenced the VGSA region. And when we then analyzed this against the phenotype for each of the mosquitoes. So what we saw was that the mosquitoes that had the mutant alleles for 976 was strongly resistant to DDT. But only the ones that had 1842 mutation were only marginally. We could not find any strong any association, but that association was only marginal. Maybe a question of maybe numbers, but none of this allele was associated with deltamethrin in this case. So none of them was conferring deltamethrin or was important for deltamethrin resistance. And so from that, then the question comes that if the KDRs are not associated with deltamethrin resistance, then what could be conferring deltamethrin resistance in this population? So another separate study we did was to just try to map the cytochrome P450s that are known to be present in Tanzania. So in this case, we focused on CYP6P, 9A and 9B. We also looked at the GST2 and some structural variants that I think is also nice to mention. That structural variants have been shown to increase resistance to pyrethroids. So in this case, just focusing on the populations that we found the KDR, we see that both the cytochrome P450s, the top ones, are fixed in these populations. And it's our thought that this could be what is driving pyrethroid resistance and making the importance of KDR or reducing the importance of KDR to the mosquito. So this could be the what the mosquito are dependent on. And just to show an example of this is this work that was done by Gareth Whittle. What we see, or what they showed here, is that when a mosquito has the resistance, the resistant, the resistant alleles for CYP6B9A, then they survive the insecticide, but they also successfully blood feed. And this just shows that this is really important for them in terms of resistance. Okay, so then the other part we had with this is now to try to form some hypotheses on where could the KDR be coming from, because it was a surprise and it's quite uncommon in Anopheles funestans. And so we had three sets of hypotheses. One of the hypotheses we had here was, could this be coming from agriculture? So we know DDT has been used for a lot in agriculture. Tanzania is quite an agricultural country. And the map here just shows the different agricultural zones, mostly the ones in dark blue. So those are the agricultural zones. So the agricultural zones are quite widespread. And the red dots on there, these are the points that we got at least appreciable frequencies of KDR. And you see, yes, they do fall within areas where you expect agriculture to be. But then because we sampled beyond these areas, and our thought is, if agriculture was really driving DDT, the emergence of KDR or use of DDT in agriculture, then it's possible that we should be seeing KDR in populations beyond what we were seeing in this study. So the other hypothesis we had was the DDT for vector control. So in the year 2008, the Tanzania NMCP adopted, and this was in response to increasing malaria cases. So they changed strategy, the vector control strategy, to adopt the implementation of DDT for IRS in areas that had persistent malaria. And they with this, they also had like a pyrethroid that was being used for the in an insect resistance management strategy. But when we checked on this, so this strategy wasn't implemented in all districts. It was only implemented in a few districts and then abandoned. But again, well, we thought that this was probably not enough to drive the emergence of KDR, which was particular to Morogoro. And I should also mention that even Morogoro was not part of this strategy that was adopted by the NMCP back in the years. So another theory that we had, and probably our most what we feel like could be the most plausible one, at least for now, is the DDT stockpiling. So again, the Tanzania government imported a lot of DDT stocks, mostly being used for agriculture, so cotton farming. But a lot of this DDT was also being used for vector control, as I had mentioned previously. So on this map on the left, this shows the areas where the DDT stockpiles were kept. And a lot of these stockpiles were kept in stores, mostly so the size of this shows the size of the stockpiles, and these are in kgs, and some of them had over 200,000 kilograms of DDT kept. And this was, I should mention that this was stockpiles that were left after what was used. So when DDT was banned in Tanzania, they did an inventory of all the stockpiles, and these are the numbers of what was left after use. So then with the Stockholm Convention coming into place, there was need to get rid of these stockpiles. And a lot of them were poorly kept, so they had leaked out into the environment. So what was done was the World Bank came in and gave some money to help to remove the stockpiles of DDT. So what happened is they were digging the soil. So the picture at the bottom there. So they're digging the soil, putting them into bags. These were put into ships and sent to the US, mostly in the US or to Europe for incineration. It was a very expensive process. And reading some of the reports from this it wasn't really successful at all because it was so expensive. And at one point they just abandoned it. So some of these DDT stockpiles are lying somewhere in Tanzania. And we believe that the effect of this into the environment could have selected for the emergence of KDR in Anopheles funestans. And this is very coincidental because in all areas that we got KDR were areas where they had the stockpiles. So then what is the KDR that we got here likely to threaten control in Tanzania? From our bioassay data, so we did bioassays with PBO, and what we see that even in populations where you have KDR, PBO still reverses resistance, and this just shows that PBO nets are still very good options to manage resistance in most of Tanzania. And this the map here just shows the trends in bednet adoption in Tanzania, and Tanzania is now adopting the use of pyrrole and PBO nets and reducing the use of either the standard nets. So what we kind of foresee with this is with the increasing use of PBO nets, then because mosquitoes really adapt quickly to what is they're subjected to, we believe that with time, the mosquitoes might kind of start now adopting and see the importance of KDR in that, and that might be enable these KDR frequencies to peak. So we believe this is it's really important to monitor this KDR, and especially its evolution in places where we've seen it, but also spread beyond what we've seen in this work. Okay, so the other aspect of this is really how you can then use genomics to monitor the impact of pollution, because I believe that if it wasn't for use of genomics and deep sequencing in this place, in this kind of study, we could have not detected these KDR mutations in the field. And I really like this quote from this book, if you can get a hold of it if you want. But it's one of the earliest books that shows the effect of using pesticides on the environment, very unintended effects. These are effects that can go beyond just biodiversity and into even human health. Okay, so I think with that, I'll stop. And there are a lot of people to thank here. My supervisors are colleagues from the Ifakara Health Institute, the University of Glasgow, the Sanger Institute, the Liverpool School, Malaria Gen, the Kenya Medical Research Institute in Kenya. And also a lot of my work has been funded by the Gates Foundation, but I also want to thank our community members. Thank you so much.
Okay. Thank you so much, Joel. That was brilliant and informative and engaging. We really appreciate it. It's now time for question and answer time, as well as discussion. So if you have a question, kindly type it in the Q&A box or type it in the chat box or raise your hand and we'll give you an opportunity to ask your question. We have about 20 minutes to go for questions and answers, so we have a lot of time. I see someone, Richard Pearson, Dr. Richard Pearson is asking, what do the frequencies of these mutations, for example, L976F, P1842S, look like outside Tanzania? I'm wondering whether Tanzania is the source of these mutations or whether they are coming from elsewhere. Joel?
So these mutations, they are very localized at the moment. They're very particular to Morogoro. I believe if someone had the opportunity to read the recent document from John Farrell, I think really simplified what we found from this. But the mutations are very localized to Morogoro. And our data is showing that it's spreading, at least in 2 locations now. But even in Tanzania, we have data from 9 other regions, but we don't see it. And the other thing we want to do is to, if indeed the mutations were rising in response to the DDT stockpiles, then it's very important to do collections around this stockpiles. So that's why we've mapped them out, and this work that we're planning to do to go around these stockpiles and see if indeed this is what is driving that. And if it is that, then I think even countries that had these stockpiles, it's really nice to have a look at that if the vectors are present.
Thank you so much, Joel. There's a comment from Jane Mwangi. Great work. Congrats on the preprint, Joel, and really nice talk. There's a question from Deo Akanbi. What is the mechanism of spreading of the KDR mutations?
Sorry, what is what?
What is the mechanism of spreading of the KDR mutations?
All right, so the KDR mutation, I think like any other mutation in the mosquito, they usually spread across generations. So they can spread through from the mother to the offsprings, and that's how they will spread through generations. And they usually within the same generation, they'll spread through what we call sexual crossing.
Okay. Thank you so much, Joel. John Farrow shared a link to the blog that Joel was referring to. It was written by our colleague, Luke Lithgow. So please go through the Malaria Gen website or just click on the link that is shared in the chat right now. Sabine Milong says, really interesting work. Great presentation, Dr. Joel. I have 2 questions of my own while I'm waiting for others to ask. I hope that I'm sure you've answered them, but just if you haven't, please do engage me. So I think one of the things you said was the high DDT phenotype resistance in Tanzania, particularly in Morogoro, was unexpected. So my question is, why was that high DDT phenotype resistance unexpected? And why was it uncommon? Is it the location? Does it have to do with genes? What is the reason for that unexpected finding?
Yeah, so DDT resistance, I mentioned, is quite unheard of or very rare in mostly in Southern Africa. This is the reason why DDT has been used a lot in areas where you have resistance to pyrethroids. But when we were doing our bioassays across Tanzania, we were quite kind of shocked to find this particular site where it's the only site where you get DDT resistance. And looking back into literature, we found one of our colleagues from Ifakara, that is Emmanuel Kaindo, also found very strong DDT resistance back in 2015. So it's kind of very uncommon that in a country, across the country, you do bioassays where the vectors are present, but only one particular place is where you find DDT resistance. So that's why we call it uncommon or unexpected.
Thank you for that, Joel. There's multiple questions coming in. There's one from Messi Tebamifo, and the question is, or it's 2 questions actually. What generation of mosquitoes did you work with? And great presentation, Joel. What phenotypic assay did you use to ascertain resistance before the molecular work? That's from Messi.
Yeah, good question. I was hoping you don't ask that, but the thing is, I mentioned in the past that the reason why we don't have a lot of bioassay data for Anopheles funestas are two. So the mosquito is very difficult to run in the lab. And secondly, finding its breeding habitats is very difficult. So the WHO usually recommends that you'll go to the field, either find immature stages of the mosquitoes in like, and then rear these to adults, and then use this about 3-year-old or 3 to 5 day old to day old, I should say, to do your bioassays. This is very difficult with Anopheles funestas. I believe anyone who has worked in the field will know this. So what we usually do is then to adopt another strategy. Some people would go collect blood-fed ones and then try to get generations from them. It's also a very slow process and also difficult. But what we usually do is to get young mosquitoes. And young mosquitoes, I mean, putting traps in areas where you collect younger mosquitoes where they come to host feed. So these are non-blood-fed mosquitoes, wild, and then expose this to insecticides. So this is sometimes it's not the recommended approach, but this is the approach that gives you the actual resistance profile of vectors in the field, because these are field mosquitoes. Should say that, yeah.
Okay, thank you so much. Methodius Okouzi says, great Joel and team. Sarah Nyasende is asking, I'm asking whether you found KDR 1014F or 1014S mutations linked to the pyrethrum resistance. That's from Sarah.
Okay, so I mean, KDR 1014, I'm not sure this is the numbering for Musca domestica. Usually it's 995 if you check on it in Gambi, or 976 in this case for fluences. But we did not find any association with pyrethroid resistance, at least for now. And for delta methrin, I should say. There are multiple other pyrethroids that are being used for vector control. We have permethrin, alpha-cypermethrin, and everything. One of the recommendations we have is to try to do bioassays with this and see if they are actually correlated with the other pyrethroids, which we believe might change the story.
Thank you so much, Joel. Stravinsky Terence Busugu has a question. Except the proximity with the occurrence of KDR mutations with DDT stockpiles, what else could have driven this resistance? Especially as I guess pyrethroids should be the insecticides that are widely used in vector control. Joel?
Yeah. What else could drive the emergence of the KDR? So, I mean, pyrethroids are widely used, I agree, even across the country. But the reasons or the theories that we've given, these are just theories at the moment. They're not confirmed. We don't have like any direct link to this. But they are reasonable kind of theories that we think they are worth following through to see on the emergence of this. So I don't think I have an answer to exactly what drives or drove the emergence of KDR in these populations, but it's something worth following, and hopefully we might have a clear answer about it in the future. But I should mention that even in Anopheles gambi, KDR can only arise in response to an intervention and an insecticide-based intervention. So it's definitely from coming from insecticide pressure, but we just don't know if it's vector control or the stockpiles or agriculture.
Okay, thank you so much. I'm switching to the chat. Diokri Matia is asking, don't you think that the higher frequency of such KDR in Morogoro could be due to large sample size you have collected in Morogoro and long sampling in that area compared to those other areas?
Yeah, that's true. And a very valid, a very valid concern or observation. Yes, in Morogoro we had kind of data over years, and this is sorry, This is because we worked in the Kilombero Valley a lot. So we get samples there easily. Outside that, we can only go to the field and do like sampling of a few days or so in other areas. So I think it's very important to also keep on sampling. And one of the things we are doing with this work is to put out a very cheap assay that you can use to genotype the KDR in the field. And this allows for more throughput kind of processing, process more samples in a cheaper way. And this will help to map if KDR is present in all other locations that we haven't sampled.
Okay, thank you so much. There's a few more questions coming in, and
Okay.
I hope we can cover them. Edmond Bernard is asking, what resistance intensity does the Anopheles funestas have? What resistance intensity does the Anopheles funestas have?
Okay, so in terms of pyrethroid, so this the data we have from our bioassays is the resistance intensity for delta methylene is very strong. What they consider if using the high resistance intensity is the right word to use. You might confirm that. We do have a preprint on this that is getting into details of this resistance intensity for the different populations in Tanzania, but it's mostly on pyrethroids. For other insecticides like bendiocab or even pyrimidine methyl, we don't see any resistance on them. So the high intensity is only for pyrethroid.
Thank you so much, Joel. A question from Faisal Ahmed. What are your recommendations for future research?
You should be telling me that, but I think one of the recommendation is to do some follow-up work that I was suggesting to go around the DDT stockpiles in Tanzania to see if it's what is driving resistance. Most importantly also is to monitor the frequencies of KDR in populations that already have it and see how that will be evolving over the years. I said like there is a change in strategy for bednet distribution. So there are new nets coming in and seeing how mosquitoes respond to this and how it affects the KDR. But there are also other things to do here. You could do like some mechanistic kind of studies, do some expressions to see on why KDR is not conferring the strong resistance that it does for Gambi, and why it's not doing that in Anopheles funesta. So these are different ways to follow through, and I'm really, really, really happy to discuss with anyone who is interested and has ideas on how to take this forward.
Interesting. Thanks, Joe. From Jen Mwangi, how might the observed decline in the frequency of KDR mutations in Anopheles funestas population between 2017 and 2023 be linked to changes in insecticide usage or environmental factors? And what implications does this decline have for malaria control efforts in Eastern and Southern Africa? That's a mouthful, but Joel?
So the decline, we believe, and also no direct correlation, of course, is that the decline in frequencies in these areas might be due to or a response to the cleanup that was being done by the World Bank through the that I just mentioned. But also the decline can also be in a response to sampling. So because you don't sample the same village every year, you'll be sampling multiple other villages. So the frequencies might maybe see that, but if it's that, then it probably won't be consistently declining over the years. So we believe this is probably a response to the cleanup exercise that was being done. And how it's sorry, what was the last part of the question?
And what implications does this decline have for malaria control efforts in East and South Africa?
I mean, let's not even push it beyond this, beyond Morogoro for now, because we don't even know the implication of the KDR for malaria control yet. So I would see a scenario that if this was to pick up in these vectors, then that's a concern. But even if it doesn't go beyond Morogoro, even beyond Tanzania, this gives a good opportunity to do some studies on its emergence or a general evolution of resistance in this vector.
Okay. As your timekeeper, I'm looking at the time. We have about 7 minutes left.
Okay.
And the questions are not stopping, which is good. It shows, Joel, your presentation was brilliant and engaging and interesting, but I doubt we'll be able to answer all your questions, so I apologize in advance. I'll still read some of them, and I hope that Joel can cover them. But for those of you that don't get to be answered now, do engage with Joel afterwards. There's a question from Ambrose Oruni, which goes, have you followed up this work by looking at if there was or if there is KDR gene flow in the neighboring countries, especially South and Democratic Republic of Congo? That's from Ambrose.
Yeah, so just quickly on that, this is the beauty of Malaria, because we do have an office from multiple other countries. So this is something that is quite easy to have a look at. And I think from what is there for the slide I presented from Mara's work is there's no KDR beyond Tanzania. We don't see it in any other country. So I think doing a gene flow will be another thing, but gene flow based on KDR probably not possible at the moment.
Okay, so there's another question from Dr. Dayo Akanbi. Congratulations once again. Joel, do you think there should be concern about resistance to other currently used insecticides, such as pyrethroids and other organophosphates, such as cypermethrin, deltamethrin, etc.?
I would say yes or no. And if concern you mean in terms of the KDR, then yeah, I mean, knowing what KDR, how KDR really works with the in the Anopheles gambi complex, then it should be a big concern, and it's something that is worth putting some attention to monitoring. It's how it evolves with time.
A question from Nila Zazai. They're asking, do you have a collaboration with NYU Professor Youssef? He's working on malaria in African population. I think the name is Professor Youssef Idagado. Any response from you, Joel?
The short answer, no. But yeah, happy to speak to people.
As I said, they're setting you up for connections
That'll be great.
networking. So, a question from Becca Love. Do you know which of the two mutations arose first?
Good. That's a very good question, actually, and I'm really hoping someone like Chris might chime into this, but it's we haven't done the analysis to show on to try to check on that, but I think it's probably the 976, and then the other mutation comes after it. Chris, do you have any indication on how this happened in Anopheles gambi?
Apologies, I have to admit, I was just replying to an email. Could you repeat the question?
Now, I think one of the participants is asking, so the two KDR mutations that we have in Anopheles funescens, which one of them arose first? And I was just thinking, how did it happen in Anopheles gambi?
So we've never really dated them. It's kind of quite difficult. We've got, as you showed in those lovely haplotype networks that someone made. There looks like there's multiple origins of these mutations, and then they spread very quickly, and you get a lot of mutations on those backgrounds. So it should be possible to try and date them. It's something that we'd really like to do. It's something that we kind of always wanted to do, and try and track them back to where we think they evolved, and see if we can understand whether there's something special or important about where these things evolved. But it's not something that we've been able to do yet.
Okay, thank you.
Thank you so much, Dr. Clarkson. And thank you so much, Joel. I'm looking at the time, as I said, so I'm going to ask one last question, and I apologize to everyone who has more questions. This is again from Jen Mwangi, who's asking, what are the potential consequences of the strong association between the L976F KDR mutation and survivorship to DDT exposure in Anopheles funestus populations, especially considering the ban on DDT in Tanzania and the lack of pre-qualified DDT products by the World Health Organization for vector control? Joel?
So I think if you are to base this on DDT, then you might be missing the bigger picture. So yeah, DDT is not currently used for control, but DDT and pyrethroid resistance are really, they always go together. And it's because the resistance to them are usually through the same the same target site. But it's only that for pyrethroids, they have an additional benefit of the P450s, which is P450s are kind of fixed in most populations across Africa now. And so if you look at this just in terms of DDT, maybe you might not see, but we believe that this is actually going to change soon, and these KDRs might be really important for other pyrethroids in the short term. But also, I think a lot of these studies you can't do. I believe if you had like a lot of mosquitoes exposed and do association studies with them, there might be a chance that you could start to see some strong indications with that. But yeah, these are follow-ups that we are planning to do, and hopefully we can provide updates on. Thanks, Jen.
Thank you so much, Joel. This was phenomenal. It was a brilliant presentation. Congratulations on the preprint. We really appreciate your time. We are out of time, so I'm sorry to those again that couldn't get their questions answered. However, you can follow Joel on Twitter. His handle is @Odero_Joe, or you can follow him or invite him on LinkedIn, and you can start engaging from there. And potentially you can collaborate on future projects, etc. Joel, thank you so much for your time. I know you're very busy. We really appreciate being educated by you, and we appreciate you taking time out to give this really amazing presentation. To all of you, thank you so much for attending today's Journal Club. There's going to be a second one on the 28th of May. So be on the lookout on the Malaria Gen GSU social media sites, where we'll be advertising the next webinar. But for now, thank you so much. If you want to go back to watching the webinar, it's going to be up on our YouTube channel at some point this week. And then you can share with your networks as well. Thank you so much, everyone. This was an excellent presentation. Much appreciated. Goodbye.
Thank you, Pablo. My pleasure.
Thanks, Joel.
Thank you.